So welcome everybody, and thank you all for joining us today at the second quantitative seminar of winter quarter. My name is Abby Bratt, and I'm one of the organizers of this year's quantitative seminar series from the Converse Lab. And I'll begin with a quick land acknowledgement. The University of Washington acknowledges the Coast Salish peoples of this land, the land which touches the shared waters of all bands and tribes in the Suquamish, Tulalip, and Muckleshoot nations. Our lab also acknowledges that we live and work on the ancestral lands of the Duwamish people, the first people of Seattle. So our presenter today is Dr. Kiona Ogle. Kiona Ogle is a professor in the School of Informatics, Computing, and Cyber Systems at Northern Arizona University, and the program director for the NSF NRT-funded T3 program that is providing interdisciplinary graduate training that integrates informatics, ecology, and team science approaches. Her research uses advanced statistical tools in an integrated framework to develop a mechanistic understanding of carbon and water cycling in arid and semi-arid systems, as well as soil carbon dynamics and the role of antecedent conditions in governing ecological responses, or ecological memory, especially in the context of plant responses to environmental perturbations. Dr. Ogle's training was similarly interdisciplinary with postgraduate degrees in biology and statistics from Duke University. So Dr. Ogle will take questions after the talk. And just as a reminder, you can submit those questions in the question box, or you can raise your hand and I can unmute you at the end. So thanks everyone for coming, and a big thank you to our speaker for sharing your work with us. And with that, it's my pleasure to turn it over to Kiona.
Thank you, and thank you for the invitation, for the especially to the seminar organizers. The last time I was actually at University of Washington was many years ago when I was a postdoc, so it's kind of a bummer I don't get to go there, but it's so much more convenient to talk to you remotely though. So what I want to talk to you today about is this title, Lags and Legacies, specifically temporal features of climatic influences on plant and ecosystem processes. This is a general theme in my research group that many of my students and postdocs are working on. And for today's talk, I do want to acknowledge some primary contributors to the work I'll summarize today, including Drew Peltier. Who is a former PhD student in my lab group and stuck around for a little time as a postdoc, and now he's moving on. And then also Jessica Guo, who you can't see in the background. She was also a PhD student in my lab. And a lot of the statistical modeling work was done in collaboration with Jarrett Barber, who is a faculty and statistician in my department here. So I want to acknowledge them up front. There's a lot of their contributions in today's talk. So I want to first give an outline just so you know where I'm going today. First, I'm going to give a simple overview of some of the motivating questions related to this concept of lags, legacies, and ecological memory. So I want to sort of summarize conceptually the general approach to inferring those properties, lags, memory, and legacies, and then I'll spend a lot more time giving a more detailed description of the analytical framework, which we call the SAM approach, and I'll describe what that is in a few slides from now, and then spend some time on some examples that apply that approach to real-world data that span ecological data along a pretty large temporal scale. And then I'll just end with some implementation considerations, things we have to think about if we do want to implement this type of framework. We'll start with this overview. An overarching question that plant and ecosystem ecologists such as myself are interested in is what governs plant and ecosystem functioning. I think we generally agree that exogenous factors such as those related to soils, climate, and weather, and endogenous factors representing internal plant or ecosystem feedbacks are important drivers of plant and ecosystem behavior. For example, in the arid and semi-arid ecosystems that my lab primarily studies, moisture availability is often one of the most important drivers of plant and ecosystem functioning. But the timescales over which moisture dynamics and other drivers affect plant and ecosystem processes is not entirely clear. And a typical approach or assumption to evaluating drivers of ecosystem functioning is to relate observed responses or variables, such as, let's say, carbon or water flux components, to relate those to concurrent drivers, such as soil moisture status at the time that that particular response is measured. But it's probably more likely that concurrent and antecedent or past conditions simultaneously govern plant and ecosystem functioning. Furthermore, past conditions may be even more important than current conditions, as represented by the arrow thicknesses in this diagram. For example, this big thick arrow indicates that the driving variable or environmental conditions 2 time steps ago is probably more influential than driving conditions in any other time. But how do we know the timescales over which such drivers influence the system of interest? That is, which of these arrows is essentially the thickest? Addressing that type of question may be achieved by designing experiments to cover a wide range of conditions that produce varied combinations of concurrent and antecedent conditions, and that generate response and driver time series data over long periods of time or over pretty frequent time observations. And modeling approaches generally need to be combined with such data to uncover these timescales of influence as related to memory, legacies, and lags. So I want to cover a couple of definitions to indicate the difference between how my lab group and I think about legacies versus memory. So before going into some of the actual detailed examples that illustrate the approaches to uncovering the timescales of influence, including memory, lags, and legacies, I first want to review what I mean by legacies and then memory in the next slide. So in our case, we focus on legacies of climate events. You can look at legacies of other types of events, but climate events in particular, such as drought legacies, for example. Such legacies are interpreted in the context of an extreme event, such as a severe drought. For example, if a drought event occurs at some time, for example, occurs at this particular time, in this example here, a drought legacy then describes the lingering effect of that drought on the ecological process of interest. Here, this could be tree growth, for example, or ecosystem carbon flux, some ecological response of interest. And a drought in 1 year, for example, right here, continues to impact, for example, that productivity or growth for multiple years potentially, or a long time period after that particular drought or that event has passed. So that continued impact we refer to as a legacy effect. And there's different properties of the legacy. There's this initial impact, how much does it actually impact or affect the response, and how long does it take to return to the pre-event conditions or behavior or the pre-drought behavior. So these ideas of the initial impact of the legacy, the magnitude of the legacy effect, and the duration of the legacy effect, you can also relate to common terms and ideas in ecology related to resistance and resilience of ecosystems. And in this particular example, the big figure here shows a negative legacy. For example, if this is a drought, the drought event greatly reduces growth or productivity, and it takes a while for that growth or productivity to return to that pre-drought kind of higher levels. But you could have a positive legacy, like in this panel B down here. And this could be, for example, there was a very wet year that happened in the system in this arid environment, and that extra moisture input stimulated productivity, and that moisture input could continue to affect productivity for many years or days to come. So either direction could happen depending on what kind of event we're looking at. But the legacies are usually interpreted or thought of in terms of the response and the impact of a particular extreme event on the ecosystem process of interest. We also are interested in this concept of memory, which is tightly related to this idea of legacies. But memory and temporal lags in particular, we think of in terms of how the system responds to past or antecedent conditions, regardless of whether or not there is an extreme event that happened. So it's independent of an extreme event happening. And here, climatic memory describes the influence of antecedent conditions, for example, moisture, on the current or future response of interest, such as tree growth. And if antecedent conditions, regardless again of whether an extreme event occurred, if they're important for predicting the response in the future, this means that there's a memory effect potentially in that system. And that memory effect could also represent a lag effect or include a lag effect. So this figure here qualitatively illustrates the relative importance, that's the y-axis here, of a particular environmental driver, such as soil moisture or temperature or some other driver, that's happening concurrently with the response of interest, but also how important it is further into the past. So antecedent conditions are older and older. You can see if you look at the relative importance here, there's this little blip here that indicates that the response of interest is affected by relatively recent conditions given by this relatively high relative importance of recent conditions. And then if you look further into the past, there's a second large blip here that occurs between time steps about 20 to 50 time steps into the past. That could be days or months or whatever the relevant time unit is. And this second peak is actually relatively large or taller compared to the first peak, indicating that older conditions further into the past are even more important for predicting the response now. And this kind of peaked here would be indicative of a lagged effect, a time lag between the response of interest and conditions that basically occurred some time step, 25 to 45 or so time steps ago. So we're interested in trying to figure out these kind of temporal patterns of the response, these time scales of influence that can lend insight into memory. So memory is basically how long can the system remember those antecedent conditions. This goes up to about 50 time steps ago. And if there's any particular lagged effects that we might see. So in my talk here, I'll primarily focus on work that my lab has done to quantify the importance of antecedent conditions by quantifying these antecedent weights, these importance weights, and look at those and to learn about the system with respect to memory and lagged responses. And I'll relate some of that work then also to this concept of legacies I just highlighted in the previous slide. Okay, so now I want to switch to the second main part of my talk. And for the second part, I'm going to focus more on the modeling approaches to some extent and give a more detailed overview of the SAM framework, which I'll go on the next slide, for quantifying timescales of influence and ecological memory. So this is called SAM because it refers to the stochastic antecedent modeling approach that my lab and I have developed, and we've been implementing ever since essentially we've developed this approach. It helps us understand and quantify the importance of past or antecedent conditions at different time lags. The relative importance of those conditions at different time lags, we go and saying that it's unknown. We don't know those relative importance weights, and we're going to estimate them based upon fitting some sort of model that describes a system to observed data. And so once we've estimated those relative importance weights that I demonstrated in the previous figure, it's also relative importance of antecedent conditions, and that in itself will provide insight into temporal lags, like I illustrated, ecological memory, and both of those can help us understand what governs legacy effects and can affect our interpretation of these legacy effects in our system, the effects of particular events or extreme events. So the approach that we also take involves a Bayesian statistical approach to fitting a particular model, the SAM type model, to data, often time series data. And I just want to show this graphical model. This is called a DAG here. That illustrates, in general, conceptually, the parts that go into this model, and the key pieces are associated with the SAM framework. So each of these circles and squares represents a node or quantity in the model. The blue nodes are data quantities that you would observe. The yellow nodes are what we refer to as latent processes. These are often described by equations and that represent quantities that we cannot measure directly because when we make measurements, we observe quantities with error of some sort. And then both the peach-colored nodes at top and the gray one on the bottom represent unknown parameters that we wish to estimate. And many of these parameters, especially these peach-colored ones, we refer to as process parameters, and those are parameters of real interest that will provide insight into the system, especially in terms of memory and lags in the work that we're doing. So when we pair the data that we have with the actual model, all the equations and stuff that underline this graphical model, the data model is described by this general equation here. We have some observation of our response variable at different times. So we have time series data. It follows some distribution that we specify that may be appropriate for that kind of data. For example, a normal distribution that describes the variability in that those data, the y conditional on, for example, expected value or predicted value, and probably some sort of measurement or observation error type term, such as this variance term. And then we have a process model that essentially quantifies that mu, which is the mean or expected value of that observed quantity, so the predicted tree growth, the expected tree growth at any point in time. And that's written as a function of parameters that we wish to estimate. And also it's a function of covariates. And then here again, the covariates where some of them are going to be these antecedent covariates that we're going to define. So X-ant would represent exogenous antecedent variables, such as past soil moisture. And E-ant might represent some sort of endogenous variable, such as tree age or past ring width production. And ZT here could represent other covariates in the model that are incorporated concurrently, so the antecedent approach may not apply to them and they would really affect the response concurrently. But important to the SAM framework is this definition of an antecedent variable. So X-ant here is simply described as a weighted average of past observed values of X, of that covariate. The W's here are the weights. They're these relative importance weights that tell us about how important was X at lag J to predicting or understanding the response at time T. So those importance weights tell us about the memory pattern in the system and potential lags. And that same sort of weighted averaging approach is applied to both exogenous and endogenous variables and summed over all possible time lags that we're considering, so summed over all J. And as part of the Bayesian framework, we then end by specifying prior distributions for parameters that represent a prior uncertainty or that impose constraints that must be obeyed within the modeling framework. And one example of a constraint here is that these importance weights, the W's, are kind of like probability parameters. And an individual W for any particular lag has to be between the value of 0 and 1, since it's a relative importance of that covariate at that lag J. And all the W's have to sum to 1 over all possible lag or time steps. So in order to implement that particular constraint, which it's like a mass balance constraint, we implement this Dirichlet prior, which obeys those constraints, the sum to 1 and the between 0 and 1 prior. It's kind of like a multivariate uniform distribution. That's the basic framework that we apply, and a lot of the modeling comes into describing this process model here, that function. What is that function? What are the key variables that go into it, and how do we interpret the parameters? So we've basically been using the SAM approach to analyze all sorts of data that we've collected in our lab or that have been provided to us, just because in many of the systems that we work in, antecedent conditions are definitely important to understanding system dynamics, and so we need to be able to account for them within our analysis framework to address a range of questions. And so I'm just showing you here on this diagram the types of data and processes that we have analyzed by this framework, going from very kind of fast time scale, things that vary in the order of minutes to hours within a day, to processes that very much more slowly, such as annual tree growth or annual forest productivity. And these are mostly from arid and semi-arid systems in the Western US. And for today, what I want to do is just give 2 examples to illustrate the kinds of things that we learn and the SAM approach. The first example will focus on these fast timescale responses and look at some plant physiological data. And then the second example I'll give focuses on a much slower dynamic process related tree growth and forest productivity that varies in an annual timescale over hundreds of years. So let's begin with that first example. This is focusing on data that describe what I refer to as plant hydraulic behavior or plant water stress. And in particular, the data I'll talk about today is data that Jessica Guo and my graduate students collected on Laria tridentata or creosote bush, which is a widespread desert shrub in the warm and hot deserts of North America. And we're interested in understanding what regulates the exchange of carbon and water between the leaf and the atmosphere. That's very important in these arid and semi-arid systems where plants can be extremely water stressed, and that can affect their ability to take up CO2 and photosynthesize and eventually grow or survive. So when I talk about hydraulic behavior, we're looking at processes that describe how leaves regulate water loss. And on the leaves, there are the stomata, and they're specialized cells that can open and close. So there's basically apertures or holes in the leaf, for those of you not familiar with stomata, but they regulate the loss of water and the uptake of CO2. And that in turn is controlled by plant water status, and it also can feed back to affect the plant water status, so how water stressed is the plant. And we're interested in how the hydraulic behavior, how the stomata respond to environmental conditions such as air temperature, deep and shallow soil moisture, where the plants have access to moisture given where their roots are, and also vapor pressure deficit, which describes the dryness of the atmosphere, which really has a strong effect on regulating stomatal behavior. So we're interested in understanding temporal variability of that hydraulic behavior and understanding what factors govern it and over what time scales do they govern that, the model behavior. So Jessica collected continuous data on plant water status. This is a very unique data set using instruments that aren't commonly used, are relatively new. These are stem psychrometers here that you put on a stem. So this is not her plant. She's working on creosote bush down here. And she rotated these instruments among different branches and shrubs over 2 different growing seasons, including the intervening winter period. And those instruments record plant water status every 30 minutes. And again, she did that for over a year. And we also have accompanying weather station and soil moisture data that tell us about the environmental conditions at the site. And I just want to show you what some of those data look like. I'm just showing data for 2 different shrubs in this site, and I'm showing you the data that's actually given is plant water potential. So an index of how stressed the plants are. The more negative the number, the more water stressed they are. Basically, a value of 0 is like pure water sitting on the surface of the soil. So a value of 0 in this case is probably more of an instrument error and not really what the plant is experiencing. But we have these time series data for individual shrub. The value varies within a day just due to environmental conditions that are diurnally fluctuating and plant physiological behavior that varies over the day. And then they vary over the season across time due to drying soil, moisture inputs, changes in temperature regimes that occur seasonally. And here's just summarizing data. Again, I'm showing you now some of the weather data, the meteorological data that goes along with it. We have air temperature, so you can see the seasonal fluctuations in that. These little bars here show you when it rained. So it doesn't rain that often in the Sonoran Desert. It does rain during the monsoon period. That's our wetter period, our summer period. And it's pretty dry during the spring, pretty hot and dry. And here's the atmospheric vapor pressure deficit D. So high values mean the air is really dry. Plants are potentially stressed. The air is basically trying to suck water out of the plant, and they have to be able to control that water loss by closing their stomata. And then we have soil water at different depths at a shallow soil layer where there's probably quite a bit of roots, and also at a deeper soil layer, which is only 45 centimeters, but where we also expect the shrub to have roots. And then I'm summarizing here in the bottom panel the actual water potential data summarized across the shrubs for each day. And the dark color, dark green, represents what we refer to as a pre-dawn value. That's the water potential right before the sun comes up, and usually represents the least stress condition of the plant. So it's in the when it is the happiest during the day. And then as the day progresses and gets hot and the sun gets really intense, then we get lower and lower water potentials. We have this midday value is more negative than pre-dawn. So this just summarizes what's going on. You can see the variability across shrubs by these air bars here within a given day. But I want to use that data to understand this hydraulic behavior of the plants. And one way we could do that is based upon this simple theory developed by Martinez Villalta that says you can relate that midday value to the pre-dawn value. And for example, a particular point in this plot would be the midday value on a given day related to that happy pre-dawn value in the morning. And if you fit a line, regression line, to those points, you can get the slope from that line. Here this is represented as sigma. And that slope is a key quantity that tells us about the hydraulic behavior of the plant. If that slope is basically close to 0, that line is flat, right? So midday value doesn't matter what pre-dawn value is, it's nearly always constant. And that represents isohydric behavior, where plants regulate leaf water status. They open and close their stomata to maintain a constant water potential throughout the day. On the other hand, as sigma approaches 1 and gets larger than 1, that represents anisohydric behavior, and the plants are referred to as having loose regulation of leaf water status. They don't really open and close their stomata very much. They let their midday values drop to pretty negative numbers, but they can still maintain some physiological function. So those kind of plants would probably be pretty drought tolerant if they're able to allow their water potentials to get to really negative values. So we're going to fit this simple model to all that time series data where we have midday data and pre-dawn data for individual shrubs. And I'm going to build up the DAG that shows you how we do that and then how we build in the antecedent effects. So here we have midday. That's treated basically as the response variable, and pre-dawn is a predictor of midday. So pre-dawn is giving rise to the midday value, And then there's these parameters, the sigma and the gamma, that are the simple slope and intercept parameters in that linear model. But then we had a second layer model, or second layer regression model, that says that slope parameter, which describes that hydraulic behavior, and the intercept, which is sort of this baseline water potential value, are then themselves expressed as functions of environmental conditions. So each of them is expressed as a function, like a linear regression, on temperature, maximum temperature of the day, vapor pressure deficit, shallow soil water content, and deep soil water content. And each of those covariates goes into that regression model and is associated with coefficients or parameters that describe the effect of those covariates. So that would be the beta parameters in the model for sigma and the alpha parameters in the model for gamma. And these epsilons here represent shrub random effects, such that different shrubs may have slightly different values of that sigma term or that gamma term. So in the end, this looks like a mixed effects linear model. But what's important here is that these covariates are all antecedent quantities, and the ones that are outlined in red, for the antecedent calculation, that's a weighted average over the concurrent value and past values, whereas this T max is just a weighted average of past values. It does not include the concurrent value. And the development of this model is based upon our general understanding of what controls the physiology of these plants. And remember that each of these antecedent variables, again, is a weighted average of the actual observed values of those covariates at each time step, with these W's being the relative importance of those covariates at different times into the past. And in this model, for those importance weights in the covariates, we allowed for different initial time scales or time steps when we implement these models. So these three on the right-hand side, these three relate to water status conditions, so moisture status of the atmosphere or the soil. These are all included at daily time steps, so we're taking weighted average over the past 7 days. And then this T max is incorporated at 3-day blocks, going up to about a month into the past. So allowing us to represent longer term temperature growth conditions by averaging over the past 28 days. We implement that model with that time series data for both the pre-dawn and midday for all shrubs at the site over about 1.5 years. And remember, we're fitting this linear model overall to those data. And implemented Bayesian framework, we get out from the estimates of sigma that are going to vary over time, because sigma is modeled as a function of covariates that are varying over time. And here's a panel for sigma. The symbols here represent a point estimate, like the posterior mean. And the whiskers here are uncertainty intervals, and here they are the 95% credible intervals. And you can see how this is varying over time. And I've just showed you the scale here. The sigma, if it's 0 down here, it would be a purely isohydric plant. And as it gets more extreme, that behavior gets more anisohydric. So a couple takeaway points here is that, first of all, if you just look at that plot, the predictions of that hydraulic behavior, that sigma term, are pretty tightly constrained. The credible intervals are pretty tight, with the exception of maybe 1 or 2 periods where they're wider than the other periods. And it also shows that Laria, this particular shrub, spans a wide range of hydraulic behaviors, and we've colored them by the categorical behavior. So, and sometimes you would classify as a species with partial isohydry. On other times, you would classify it as a species having extreme anisohydric behavior. And what's really interesting about that is that the past dogma has typically assumed that a given species possesses a single type of behavior. It's either isohydric or anisohydric. But the field is generally moving away from that idea and recognizing that behavior is more dynamic. And you might ask, okay, what explains that temporal variation in that hydraulic behavior? So why is it varying so much over time and switching from these different categories? Well, the main things that are controlling this behavior are, in fact, antecedent or past moisture conditions, both the atmosphere and the soil, and temperature is also important for predicting that response, especially in terms of how the stomata are behaving. So just to give an example, these peaks where the behavior is like jumping up into anisohydric or extreme isohydric, those are periods where the deeper soil experienced a moisture pull. So deeper soil is relatively wet, even though shallow soil could still be relatively dry, or the air could be relatively dry. And if we also look at the results in terms of timescales that influence, so I said antecedent conditions are really important. Over what time period are they important? Are there any lagged effects? And here I'm showing you the relative importance weights for vapor pressure deficit, so atmospheric moisture condition, and deep soil moisture. And if you just look at these two and look at the weights and how they vary from concurrent importance all the way up to 7 days ago, you can see that yesterday's vapor pressure deficit is more important than today. So today's vapor pressure deficit doesn't really have much impact on stomatal behavior, but what happened yesterday is more important. You can't really clearly resolve deep soil moisture antecedent weights, even though deep soil moisture is important, but there's no clear lag response. There's no clear time period that's more important than other time periods. That's in contrast to looking at the importance weights for shallow soil moisture and antecedent temperature. So concurrent shallow soil moisture is very important. It's a super high weight, and past conditions of shallow soil moisture aren't nearly as important. And if you look at temperature, there's this blip, this weight that's much higher for the 14 to 16 days ago. So lag temperature about 2 weeks ago is important for predicting stomatal behavior today. And that's not surprising because that likely represents some sort of temperature acclimation response of the various enzymes and systems that are controlling how the stomata operate. So that was the first example. That was on a fast time scale. Now I'm going to move to the other end and give a quick highlight of examples how we've implemented the SAM framework to tree growth and forest productivity, representing productivity on an annual time step. So some of the general research questions that we're interested in when we focus on these data are what climate factors govern annual tree growth and forest productivity in general? Over what timescales do these factors influence growth? How long does a particular climate event continue to influence growth? That's a legacy effect question. How do the answers to these questions differ among species, and how do they vary across space, such as along environmental or climate gradients? And the approach to addressing these kind of questions is to utilize tree rings. Tree rings are really cool resources, tools. You can take this core from a tree using an increment borer, much like a drill, and you sand it and prep it. And every one of these rings here, these bands, represents, the width of that represents the annual vegetative growth of that tree. And you can get hundreds of years of growth on an individual tree. And when you measure those, you can also use various techniques to determine what year was that ring produced. So you can relate ring width to a particular year, and then you can go and look at the climate conditions associated with that year. So that's the main approach we're using to address these types of questions. I'm going to build up for you that SAM approach that we use, the general framework for working with tree ring data, although we might slightly modify it depending on what data we're working with and what question we're trying to address. But the center of this model is a response variable, ring width, that we measure for every year for individual trees at a bunch of different sites potentially. So these are individual ring widths. And then first of all, ring widths are known to be affected by tree age. So we incorporate an age effect or tree size. So as a tree gets bigger and bigger, the circumference of the trunk gets larger and the rings tend to get thinner and thinner, even though the total amount of wood put on is still pretty large because it's a larger circumference. And then there's also a strong autocorrelation effect associated with ring width data. So incorporating an effect of prior ring width helps to explain variability in ring width over time. And then each of these coverts is associated with coefficients or parameters that describe the effective age and the autoregressive effect. These parameters we often are going to estimate at the level of a tree. And then we build a hierarchical model that assumes that trees are nested within populations, such as a bunch of trees at a given site. And those populations are nested within a species. So we might have a bunch of ponderosa pine across the Southwest, and each population gets its own parameter, but those parameters come from the overall ponderosa population of parameters. So that's that one side. Basically, this is the endogenous part of the model that incorporates ring widths and age. And then we also build in the climate variables. And here, just as a simple example, we're using precipitation and temperature, which always emerge as being really important predictors, but we could include other variables, and we've tried them related to drought indices, moisture, atmospheric moisture indices. We have these climate data that are observed at the monthly timescale associated with each of the years that we have ring widths. And then we calculate the antecedent precipitation, antecedent temperature, using that same framework where we're taking weighted average over each month and over past years to try to figure out how important is climate during the different months and different past years, including the year of ring formation, so the year of growth. And then we're estimating those importance weights. And then those antecedent covariates that we're calculating are put into the model for ring width, and each of those covariates themselves is associated with their own climate effect parameters, so regression coefficients. And those, again, may be estimated at the level of tree and modeled hierarchically by population or species, for example. So that's the general framework that we use. We're really interested in estimating these weights to learn about lags and memory. And we're also interested in estimating these coefficients to learn about climate sensitivity, for example. So I just want to go through this brief proof of concept and then summarize some results from application to understanding the legacy effects of La Nina events. So this proof of concept was applied to ring width data from 7 different sites in Arizona and Colorado total across 3 different species. And we fit that type of SAM model, and I'm going to first show you an example of estimating annual importance weights of climate. So the top panel will be importance weights for precipitation, bottom for temperature. We have the importance of conditions during the year of ring formation, so year 0, and up to 4 years ago. And I'm just showing it for a collection of different species and sites. It doesn't really matter if you know what species these are. I want to just highlight some general features. One thing, if you just stare at that figure, you'll see that the temporal patterns and time lags appear to differ between climate variable species and sites. And in particular, if you look at the precipitation, current year precipitation, and maybe the previous year has the highest weights. So current precipitation is most important for predicting ring widths. That is in contrast to temperature, where if you look at the importance of current temperature during the year of ring formation, its weight is almost 0 in most of the sites except this one. So for temperature conditions further into the past, such as last year and up to 4 years ago, are important, but generally concurrent conditions are not as important for predicting tree growth. And you can look at the importance of monthly conditions, so you could try to identify the importance of different seasons. And that's what we're doing here for particular species and sites, looking at the monthly antecedent importance weights for precipitation. Going from here, this block of 12 weights is for the concurrent year, the year that the ring was formed, prior year and up to 4 years ago. And you can see how these weights are going up and down. And I want to point out a couple of patterns here. There's a blip, high weight right here in June. So that pre-monsoon, dry June conditions, the conditions during that June period are really important for predicting how much the tree grows during the growing season. Also, what's really important is this whole period, including the prior late summer up to early spring, and especially that winter period. The conditions during that period are really important for predicting growth during the subsequent growing period. And there's also some blips further in the past, including this winter a couple years ago and summer, indicating that conditions further up to 2 years ago continue to be important for tree growth. It's interesting if you compare those precipitation weights, monthly weights against the temperature weights. And again, remember that temperature during the year of ring formation was not very important, and you can see the weights are all really small for that year, all the monthly weights. And temperature has a really strong weight right here, which says that the conditions during the previous summer are super important for predicting how much the tree goes the following growing season. And it's we have this climate data on the same time scale, but both of these climate variables are affecting tree growth really differently, it appears, and different time periods are really important for predicting growth, whether you look at precipitation or temperature. So using that same approach to then approach questions that are more broader scale across more sites and more species, I want to highlight one particular project, again, that looks at the legacy effects of La Nina events. And a couple questions here that we're addressing is how does climate sensitivity, that's the climate coefficients, the betas, the regression coefficients, and growth characteristics differ in La Nina years. La Nina years in the Southwest are described as dry winter periods. So you think of this as like a dry period. We also want to know what is the importance of timing of climate events during these La Nina periods relative to sort of more normal or average periods. To address these types of questions, we utilize tree ring data from many different sites. And for example, here's data that we have for pinyon pine. We have data from 101 sites across the Western US, mostly in the Southwest. We also have data for ponderosa pine and Doug fir for even more sites. We have over 300, almost 400 sites total across these 3 species for which we have growth data. We downloaded raw tree ring data from this international tree ring data bank, so we didn't collect this data ourselves. We use that data then to detrend the data for age, so remove the age effect, and then produce what are called stand level chronology, so that at each of these points, each site, we have one time series that tells us about the relative growth of trees at that particular site or that population. And then we have it for all of these sites, almost 400. And we want to, again, focus on these questions centered around La Nina periods. And we define La Nina periods as the year of a La Nina event, so that year that included that dry winter and the 4 years following La Nina year. And the reason why we chose that 4 years goes back to this concept of drought legacies. Another previous study I was involved in that also synthesized tree ring data from around the world, in this case, found that drought events can have a legacy, they have a lasting effect, and it's specifically for conifers and arid and semi-arid systems such as the Southwest. A drought in one particular year can affect growth for up to 4 years later. So it takes about 4 years for trees to recover from that drought event and to return to their pre-drought growth. So that's how can we use that La Nina plus 4 years afterwards to define that La Nina period. And then all other years that don't fall into that category are referred to as other periods, so wetter periods. We fit the SAM model to those data, and we allow the parameters in those models to differ depending whether it's a La Nina period or an other wetter period. And this is showing the relative importance of a particular climate variable. Here it's a drought index during the year of ring formation up to 4 years ago for the 3 species of focus. And I'm separating the symbols by whether that's associated with La Nina periods or with other periods. And if the symbols are grayed out, that means there's no significant difference between the 2 different periods, La Nina and other, but when they're dark, that indicates a difference. So we can highlight a couple differences here. Actually, I'll bring up the points down here. So tree growth is more strongly coupled to current conditions. So during the year of ring formation, when it's a drought period, a La Nina period, it's trees have short memory and they're responding to conditions during the year of growth. During these more normal or wetter years, drought conditions further into the past, up to 4 years ago, are more important than you would expect given La Nina years, or La Nina years. So past drought severity is important during wetter periods. And if you those monthly importance weights and put everything together, the take-home message we got from this, if you look at sort of the typical site or climate diagram for this region, it looks at temperature, average monthly temperature over the 12-month period, going from the start of the growing season through the winter until the end. Temperature rises highest in the summer, and the blue line is the precipitation. So precipitation drops below temperature, low precipitation, but rising temperatures during this June period. That again is the dry pre-monsoon period, so before the summer monsoons. That period is really important. And we found that during these dry La Nina periods, tree growth is really sensitive to the conditions during that particular spring or May season. Such that if by chance there happened to be rain started early, so the monsoon started early, that could rescue trees from the La Nina drought. It could bring their growth back up. But if that June period, that May-June period is really dry, those trees could be even more greatly impacted. The work done with these tree ring data across a bunch of different sites has led us to think more about tree growth sensitivity to climate. And here's an example time series that shows growth patterns over time, like you might expect in general. But then there could be some stress event that occurs, such as a drought. And then there's this recovery period. And so the typical idea about how trees responded to drought, years ago was a stationary response, that the growth response to precipitation was the same over time, that sensitivity or slope did not change. And you would have predicted trees to grow along that dark line maybe during that time period. But that understanding is being updated. One example would be to think of it more in terms of homeostatic sensitivity, where during fairly good conditions, trees have a certain sensitivity, and then during drought or bad conditions, they have a reduced sensitivity, for example. So you can have a target sensitivity, then it's affected by that stress event, and then a return to a target sensitivity. What we believe is probably more realistic is that the sensitivity to climate is just varying over time. There's no target necessarily. There's no switching between like baseline and recovery, but it just changes over time depending upon antecedent conditions and can help us explain tree growth over time. Okay, so basically I'm at the end of that, and I just wanted to highlight a few things to think about if you were ever interested in implementing these SAM type models to time series data. They've been implemented to a variety types of data, including things like kelp dynamics and coral reef processing. So not just tree growth in semi-arid systems, but if you want to implement it, we have to think about when can we actually interpret those importance weights. You can't just jump in and interpret them right away. There's some conditions you have to look at. What do you do when you have autocorrelation in predictor variables? What do you do if you have correlated predictor variables? We've addressed both of those questions in many of the models that we've worked on. And there's also this potential for weak non-identifiability of effects parameters, coefficients, and importance weights. And we've developed some computational tools to help to address that, have improved what we refer to as MCMC mixing and convergence issues. So if you're familiar with Bayesian techniques, that wording will make sense to you. But if we will have time, we might come back to those, but I just want to put that up as things we have to think about. And then I just want to end again with saying, you know, this approach is really valuable. We use an analysis of all the data that we work with essentially because antecedent conditions are just extremely important. And this has proved to be a very flexible and useful approach for understanding and quantifying antecedent conditions. And with that, I'll just end and thank you. And just during my introduction, you guys already learned that I'm the director of this PhD program, the T3 program. If there's any master's students or anybody interested in pursuing a PhD in a quantitative field that's not University of Washington, you might be interested in checking out this Eco-Informatics program at NAU. And with that, I'll stop if there's time for questions, possibly.
Thanks so much. That was great. We do have about 10 minutes for questions. And I wonder, Dr. Ogle, if you wouldn't mind going back to your slide where you talk about kind of other implications.
This one?
The previous one. Yeah. Can you speak a little bit to the problems that you've encountered with respect to mixing and convergence? I'm just thinking, you know, that's something that comes up a lot in this fishy crowd when we're fitting these Bayesian hierarchical models.
Yeah, so the issues that are associated with the SAM model are very specific to, I don't want to like spend a ton of time on, but I just want to find the slide because I have them in here. Remember I said that the antecedent variable here is a weighted average of past observed variables, but that quantity itself is multiplying a coefficient in a regression model. So we have an unknown coefficient multiplying essentially these unknown weights. And that results in potential, what we call non-identifiability of the coefficient A and the weight W. So you'll see some what we call poor mixing or poor convergence of the A and W. And what we've done is to implement some reparameterization tricks that allow us to sample non-identifiable quantities and compute or monitor what we call quantities of interest. And this sort of approach is also used in hierarchical models, linear, Bayesian models that don't employ SAM framework, using techniques called like parameter expansion to help improve mixing convergence. If anybody's familiar with Andrew Gelman, he's written a lot about that. That's a little bit different than what we do here. And our solution here is very specific to the SAM framework because the weights are being multiplied by this coefficient. But just like, without going into detail, if anybody's taking notes and has that, they can look into parameter expansion and some of the work that Andrew Gelman has done.
Yeah, that's great. Thank you. We had a couple of questions rolled in, so I'll send them your way. So the first is from Dan Ovando. He says, maybe I missed this. Can you give a sense of the sample size for some of these models, coming back to the mixing and convergence with all those tricky params?
Sample size in terms of actual data or of MCMC samples?
Maybe both.
Yeah, so the data, actually, there's a large number of observations relative to the number of parameters. So I'm not concerned about the observation like data size, but for implementing this as Bayesian technique with MCMC methodologies for sampling, yeah, we often have to run some of these models for a long period of time, let's say like 500,000 iterations for 3 chains. So you're talking about well over 1 million samples in order to sufficiently explore the parameter space. Because if there is this sort of what's called poor mixing, the parameter chains aren't moving around very well, then they're not exploring the parameter space very quickly. So you have to let them sample for a long time. So some of these models, like with the tree ring data, which there's lots of data on lots of sites, can take 1 to 2 days to run. So you want to make sure your models is well coded and you're happy with it, and you test it out in a smaller subset before you let it go.
That's great, thanks. Jim Iannelli asks, nice talk. Long time since I took botany, but is there a reproductive response to stress periods, and is that something that is measured and could be included in the model?
I'm sure there is. I mean, we don't necessarily work with reproductive data or have any, but there definitely is work on plant phenological responses, whether it's reproduction in terms of producing flowers and cones and stuff, or whether it's producing new leaves. That's often been linked to past or antecedent climate conditions. There might be particular cues, like how many warm days in a row the plant has experienced before it will start to produce new leaves or flowers. But my lab hasn't worked with any data like that. But I'm sure if we have time series data, that you would definitely be able to find strong antecedent climate effects.
Cool. 2 folks have the same question, which is, it seems like otolith chronologies, would be an obvious application. Are you aware of groups that have done that?
I don't know. What was the chronology? It's probably
otolith. It's a fishy one.
The fish bones?
Yeah, the fish. I'm not a fish person either, but I believe it's the fish ear bone. Someone can pipe up if that's incorrect.
I don't know. I mean, I think I'm aware of like some sort of.
It's a measure of fish growth. They're very similar to tree rings, I think.
Yeah, I would think so. I mean, I'm aware of like some data from whales or something where you can measure rings or like tree rings. And I would think, as long as you have a long enough time series, the issue is whether or not you have a long enough time series. If you only have like 10 years of data, it's going to be hard to uncover the effect of like what happened 4 years ago, climate effects 4 years ago and what happened now. So you would have to have long-lived organisms unless you're measuring growth at subannual scales. But the length of the time series is important.
Right.
There might be studies, I just don't know because it's outside my field.
Sure. Okay, we've got 2 questions from Elizabeth Holmes. Have you explored response variables that are temporal weights rather than like value at time t? So that she adds, so the area under the curve of the predictor variable. And I can unmute you.
Yeah, I don't think I quite understand. I would have to say no, but I don't quite understand the question.
Okay, let me see. Oh, looks like she's offline for a moment, so maybe we can come back to her question. Dan has another question for you. What did you use to make those DAGs in the presentation? They look great.
Just PowerPoint. Sorry, nothing fancy.
That's okay. Let me see if Elizabeth Holmes is back.
Oh, if she comes on and can explain, but a couple of things. So there's our package that I was not involved in developing called EcoMem, and they still use the same idea of estimating the weights, but they constrain them more by using splines to model the weights. And I did have a postdoc many years ago that also tried something like that, but he didn't end up publishing the paper. But that's the only thing that we've explored. We've also explored allowing those weights themselves to vary over time so that maybe the relative importance of last year's climate could have changed from, let's say, like 1900 up to 2000. So we've done a little bit of that, but there's still a lot more work that could be done. But we haven't done anything that's really related, I think, to what Elizabeth was trying to ask.
Okay. Yeah, I'm not seeing her, but if Elizabeth, if you're here still and you want to raise your hand, I will unmute you. And then a couple of papers Lychee has shared, and I'll share them in the chat, a couple of applications to otoliths, RE, the earlier question.
Yeah, it'd be interesting. I mean, if the time series were long enough, I don't see why not. I mean, it's not like the SAM approach itself is unique to plant data. And like I said, I'm aware of studies that have used it with kelp and coral reefs. I was asked to review the coral reef paper, which I didn't. So I know it's out there. I just don't know the take-home story.
Sure. Okay, it looks like Elizabeth is back to explain her question.
Hi. Yeah, sorry, I dropped off just at an inopportune moment. Yeah, so my question was just kind of, since you've you've done a lot of different studies on this, I wondered if you looked at where the predictor variable, rather than saying like the value at time t, so in this case, say the tree growth rate at time t, at year t, if you looked at things where you are looking at the weighting over a number of years, in part that's sort of thinking about like there's some maybe autocorrelation, but in the not the predictor variable, I'm talking about the response variable. So the response variable, rather than looking at what it is at time t, you're thinking about some kind of integrated response. I'm also thinking about your initial slides where you were talking about this kind of legacy effect and showing like area under the curve over time.
Yeah. No, I mean, of the data that we're working with, I'm not sure if that would make sense for some of the quantities that we're looking at. But I mean, tree growth itself, annual, is integrating over time indirectly, right? Because we have measurements of tree growth within a year, like what's called the early wood growth, when it grows early in the season, when it grows later in the season. We have separate measurements of that, which we've analyzed separately. And then we have the annual growth. So that's like just the measurement itself is integrating over the whole year. But we haven't done anything else really like what you're suggesting. I can't think of a specific example with our data where we would do that, but I could see there maybe being other types of data that could be interesting.
Awesome. Thank you so much. I'm not seeing any more questions, and we're just oh, one more question. Sorry, if you have a moment.
Mm-hmm.
So Mark Sherrill asks, given the often hierarchical nature of the models you're fitting, how are you selecting the appropriate number of lags to include for the endogenous or exogenous covariates, i.e. what form of model selection?
Yeah, that's a really good question. And it's took us long. I mean, that also took us quite a while to figure it out. It's more like trial and error. It wasn't necessarily a formal model selection, but the more lags you add, so the more weights, then the more difficult they are to estimate, and you get potentially more computational and mixing issues. So like just qualitatively, we might find that as we add more lags, that the weights for lags further in the past just become less and less precise, and like wider and wider credible intervals. We can't estimate them directly. So we did a lot of trial and error for various data sets to figure out, hey, maybe we shouldn't be using like temperature today and yesterday. We should be averaging temperature over these 3 days and 3 days before and 3 days before that. And we kind of ended up optimizing the number of lags, but it was not like a formal process. It was more like over a period of a couple of years, we arrived at, okay, this is the appropriate number of lags for this particular type of data. And for tree ring data, yeah, I mean, I'm sorry, I don't have like, yeah, we did this really rigorously. It was more about just, yeah, trying things out and making sure that we had models that were over-parameterized and that we can interpret the weights and the parameters.
Great. He has a short follow-up question to that. Is it necessary for the sum of the weights to equal 0 for identifiability?
That they equal 1, how they're interpreted, but we've discussed other approaches where we could maybe deviate from that, where the product might be equal to something, or yeah, it's in the approach that we're using right now, it is part of it, and the weights are interpreted as these kind of probability parameters or probability conditions 10 years ago. In fact, what's happening now, but it's very possible that the weights could be interpreted a little bit differently and do not have to sum to 1. My collaborator, the statistician Jarrett Barber, has looked into that a little bit and had some ideas for that. And I forget what the specific modeling, the approach that he was looking into, but there was a different type of constraint that was not a sum to 1 constraint, but they still had to be constrained in some way.
That's interesting. Good question, Mark. He says, thanks. All right. So now I think I'm not seeing any more questions roll in. And so with that, I want to say thank you so much to our speaker, Dr. Ogle, and you're getting, the Zoom or
the virtual class.
the virtual claps. So thanks so much, everybody, for joining us. And please join us again next week. We'll have Ed Tekwa speaking about confronting the bias in biodiversity science.
Thank you, everyone. Thank you for the invitation, and have a good weekend.
Yeah, you too. Bye-bye.
Bye.